In the hierarchy of nature, the plant comes way down the list. Plants don't appear to move. They're silent. But beneath the surface, there's a whole invisible world. With the help of time-lapse photography and new scientific research, we show some of the many complexities and mysteries within the green machine. Nothing has quite the same power to colonize an area of land as the plants. They've found their way into virtually every corner of the globe. On land, they produce 150 billion tons of new living matter every year, the most abundant life form on Earth. But they also pervade the seas. It was from the sea that they evolved, one of the first life forms on Earth. This single-celled plant, an alga, was found in rocks 3 billion years old. The unique discovery that plants made was chlorophyll, a green compound based on magnesium which could turn light into the food for all living matter. As the algae, the first plants, developed, they began to move, breathe, have sex, and eventually moved onto land. The mosses came first, then the ferns. But the most rapid evolution came when plants invented the seed. The conifers appeared, and then the plants that bore flowers. In this group there are two main types: plants with straight leaves, like the grasses, and those with complex branching patterns up the stem, like this oak. An abominable mystery was how Darwin described the evolution of flowering plants, but they've come to dominate the plant kingdom. Samples of many of them are stored in botanical herbariums like this one at the Royal Botanic Gardens in Kew. At the last count, Kew had specimens of well over 500,000 individual plants, but new ones are being discovered all the time. This is a recent candidate from Cyprus. It was Linnaeus, 3 centuries ago, who started plant classification, and today his nomenclature is still being used, and in the original language of scientific discourse.
Scylla mauricii, species nova, floribus lilacinis, campanulatis, perianthii segmenti subconventibus manifesti discedit.
A new flower has been discovered to add to the 200,000 already known.
Ovoideus, 2 to 2.5 centimeters longus.
It's another example of the diversity of the plant kingdom, even within the same botanical group. Take the genus Euphorbia. One of its more famous species is the poinsettia, but its relatives can differ widely. Adaptation to new habitats has produced a startling range of forms out of what was once a common ancestor. It's clear part of the plant world's success has been due to their methods of sexual reproduction. Unable to select their mates like animals, they've used insects or the wind to distribute their pollen. Some flowers have developed special mechanisms to make doubly sure the pollen gets dispersed. Some plants have ways of ensuring that the pollen only works when it comes from a different flower. Pollen grains and stigmas have protein recognition systems that prevent self-fertilization. In this way, plants maintain their genetic variability. The power to adapt is ensured. The strength of growing seedlings is phenomenal. It's been calculated they can exert a force of 450 pounds per square inch. But at first, most rapid growth is in the root. It can grow up to 4 inches a day, and choosing the path of least resistance, its movement downwards is inexorable. How does it do it? Put a root on its side, and within 15 minutes it'll start to redirect itself. The gravity sensing device is in the tip, because if you remove it, the root will be completely disoriented. Inside the tip, cells have been found containing large granules of starch, and it's thought these fall by gravity onto the wall of the cell and so tell the root which way is down. Starch grains like these have also been found in the shoot, but in this case gravity makes shoots respond in the opposite direction. These nodding movements made by most growing plants are believed to be subtle checks of gravitational alignment. But shoots have another way of directing growth: light. This directional response is rapid. Place a light to one side and the plant starts bending within 10 minutes. How the plant does it was first investigated 100 years ago by Darwin. He put tiny metal caps on the tips of seedlings, shone a light to one side, and found they didn't bend. And he noticed too that the untouched plants bent not at the tip, but further down the shoot. Darwin likened the tip to a primitive brain, sensing the light and then sending some influence down the shoot. What that influence is, botanists took 100 years finally to establish. They cut off the tips of literally thousands of growing plants in the hope of finding some chemical substance that could make plants bend. Extract of shoot tip was purified, and out of hundreds of substances they found one that looked like the answer. It was present in minute quantities. A paste containing the substance, applied on one side of the top of a plant, makes it bend as if towards the light. Here's another blob on top of an oat seedling. A chemical had been found in the 30s that did the same thing, but it wasn't until 5 years ago that the substance was actually found in plants. Called auxin, it's a hormone produced naturally by the plant in the tip. To make the stem bend, say, to the left, auxin moves down the right-hand side. There, it makes the cells more permeable to water. They enlarge and force the plant over. Auxin was once thought to be the only plant growth substance, but in the 1950s they found another one, gibberellin. In just 3 days, it'll have dramatic effects on growth. Cytokinin was discovered next. A millionth of a gram of it can make these tiny clumps of individual plant cells divide rapidly. Then a growth inhibitor was discovered, abscisic acid. It's naturally produced by the plant in, for example, a drought to conserve water. For the ripening of fruit, there's another hormone, this time a gas, ethylene. It seems a complex picture, but it's not the whole story. At Glasgow University, Dr. John Hillman.
The great problem concerning plant growth hormones revolve essentially around the fact that they're present in the plant tissues at such low concentrations of around 1 part in a million, and very often much less than that. So that poses instantly a very severe problem of analysis.
It's only recently that modern techniques of biochemical analysis have entered the world of botany, and they're beginning to reveal an even greater complexity. This peak is just one of the hundreds of new compounds they've discovered within the 5 groups of plant hormones. And modern analysis has upset some long-established theories. Take this familiar growth response. As every gardener knows, if you remove the growing tip of a plant, it'll rapidly compensate. These tiny buds will, within days, have got the message to grow. But how? The textbooks say that if you put a blob of the hormone auxin on the cut tip, the lateral buds will never develop. They are inhibited by auxin. But a year ago, the theory was scuppered. The Glasgow team did the first critical measurements of the hormone levels in the buds of a bean plant, and found the textbooks were wrong. The auxin levels rose rather than fell, and what's more, the buds developed very rapidly, within 20 minutes.
Our knowledge is still very much at a rudimentary level. The plant growth hormones that we know about at the moment still can't account for all the coordination mechanisms that we see in higher plants. There undoubtedly are a lot more substances yet to be discovered, and there are certainly a lot more mechanisms yet to be elucidated.
Botanists are only just beginning to appreciate the complexities of plant growth, growth that has produced the largest living structures on Earth. Trees can reach 300 feet high. This may not seem remarkable, but the theoretical problems are immense. Quite simply, how does a tree get water up from the soil to the top? The question has been investigated by using colored dyes. Intrepid botanists have sawn off trees at their base, suspended them on scaffolding in a tub of water, and seen what happens to the water. Within an hour, the dye has reached 60 feet. Though completely separated from its roots, this tree will happily convey 20 gallons a day 200 feet up into its topmost leaves, and at a speed of up to an inch a second. But how? Could it be capillary action? Liquid can be drawn up very fine tubes. Perhaps atmospheric pressure. In fact, neither would get the water very far, 32 feet at the most. The plant must have some kind of massive suction pump, and this is it.
What happens is that the sun's energy evaporates water from the leaf, and this generates a chain of suction down the whole plant because the water molecules stick to each other. It's a very special property of water, and the water molecules are lost from the top, and there's a whole kind of chain of water pulled through the plant from the soil.
This is a crude laboratory model of the system. A porous pot filled with water represents the leaf. As the water evaporates, it creates a kind of molecular suction up the tube. In the laboratory, water has been raised by this method the equivalent of only 60 feet. For it to work in trees, the leaf would have to be operating at much higher pressures. To measure the suction power of leaves, scientists have cleverly reversed the process. Inside this chamber, a leaf will be put under pressure. They'll see at what point the sap in the leaf is forced backwards down the stem. The pressure needed to counteract the suction in that leaf was 180 pounds per square inch, equal to about 15 times atmospheric pressure. This bottle gives some idea of the power involved. It explodes at 15 times atmospheric pressure, or in the parlance, 15 bars.
In a plant such as this, the suction that will be used to draw the water, only that modest distance there, is of the order -4 bars. And even this is nothing. Many plants will normally live at -30 bars. That's equivalent to a suction up a vertical tube of 300 meters of water. And that's incredible. But plants are able to do it without any problems whatsoever.
So how do plants withstand these incredible pressures? The answer lies in the structure of their plumbing system. This is a cross-section of the stem of a lime tree. We're travelling up the trunk. The holes are the pipes that take the water up. Between the tree rings, the pipes form an intricate network. Their strength comes from the way they're constructed. Some with hoops, others with net-like structures. Engineers call it geodetic. But there's another set of pipes that bring sugars down from the leaves to the roots, and the pressures here are equally high, up to 450 pounds per square inch. At intervals inside the pipes there are valves. If the plant stem is damaged and starts to leak, they'll immediately seal off the flow. And the power that drives the plant plumbing system? The sun. No one's sure why leaves come in so many different shapes, but it is known they will arrange themselves on the plant to receive the maximum amount of light. Some leaves have evolved ingenious ways of avoiding shading their colleagues below them. In harsh climates, they have the problem of loss of water through evaporation. The solution, smaller leaves. The conifer's done it, and so has the cactus. But whatever their shape, all leaves have the unique property of manufacturing food from light. Photosynthesis. It begins here. The underside of the leaf is perforated with tiny holes, 1/1000th of a millimetre wide. Through them, the plant draws in carbon dioxide from the air. It can open and close them to regulate the flow. But while the plant is drawing in carbon dioxide, it's also exhaling oxygen. Measurements of the oxygen output provided one of the first clues about how photosynthesis works. Because if you dim the light, oxygen will be cut almost immediately. And in an amount directly proportional to the amount of light. So photosynthesis must be a very precise photochemical process. But besides oxygen, the plant also makes carbohydrates. One way botanists have studied that process is to stick a film negative on a leaf that's been kept for some time in the dark, and then shine a light on it. The different densities in the film will let through varying amounts of light onto the leaf. Boil the leaf up, drop it in alcohol, and you've got a print made of carbohydrate. Photosynthesis in leaf cells has, in a way, the photochemical sensitivity of film. It was only in the 1950s that a physicist, Melvin Calvin in California, and a biochemist, Robin Hill in England, unravelled the underlying mechanism. These are photosynthesizing plant cells. To them, Calvin introduced radioactive carbon dioxide and then took samples every few seconds. He wanted to follow the stages of carbohydrate production during the early moments of photosynthesis and found their molecular structure revealed much about the chemistry of the process. Today there are over 600 laboratories specializing in the subject, and though the details are still baffling them, this is how they think it works. These are leaf cells under the microscope. Inside them there's a constant stream of green particles called chloroplasts. They do the photosynthesis. Under the electron microscope, these are chloroplasts at higher magnification. Magnified 40,000 times, the inside of the chloroplast is shown to be made up of tiny collectors of light arranged in tiers. Each collector is itself composed of thousands of molecules of chlorophyll. These trap the light and deliver the energy to a number of specialized molecules. This is one of them. Light hits it and excites an electron. The electron leaves the molecule and energizes a reaction between hydrogen and carbon dioxide. This manufactures the carbohydrates. But that's only part of the picture. In another molecule, another electron is excited to replace the first. A byproduct of that reaction adds more fuel to the carbohydrate manufacture. Meanwhile, the plant has taken water and split it into its component parts, hydrogen and oxygen. That produces another replacement electron. Just how a plant splits water isn't known. In fact, the details of photosynthesis itself are still mysterious. In an almost magical way, it can turn light, water, and a dead gas into the energy for living matter. Without it, we could not exist. But the plant's use of light doesn't end there. Take this cucumber seedling growing in the dark. The absence of light produces long, straggly growth. But just one flash of light is enough to transform it dramatically. its leaves begin to open to collect light for photosynthesis. So how does a plant actually see light? One way to find out is by breaking up light into its component wavelengths, in other words, the spectrum. Our eyes have receptors that see these wavelengths as different colors, but what about plants? Experiments have shown that plants can not only perceive different wavelengths of light, they actually use them to control their growth. This is what happens in blue light. They bend towards it. Blue is the wavelength plants use for direction. In green light, they grow as if in the dark. Plants are blind to green. Under red light, growth is more normal. So somewhere in the plant, there's both a blue and red wavelength receptor. But plants don't stop there. They can detect wavelengths of red beyond the limits of human vision. This is how plants react when longer red wavelengths are added to the visible red. They grow up to 10 times faster. These invisible wavelengths are in the region botanists call far red, and if you switch them off, growth reverts to normal. The search for the plant color receptors began 25 years ago. Green was the only color the scientists could work in. The plants couldn't see it. It's not been a total success. To date, the blue light receptor has not been found, but they've had more luck with the red. In a purified extract of plant tissue, they discovered a faint blue pigment which was extraordinarily sensitive to the longer wavelengths of light. It did a complete chemical switch as soon as the wavelength entered the far red. They called it phytochrome. Phytochrome is now believed to be a sort of botanical light meter. With it, plants can tell if they're in the shade or the light. Because under the shade of leaves, the wavelengths that penetrate most are the far red. Phytochrome detects them, it switches, and growth speeds up, taking the plant rapidly nearer the light. Once in more normal light, phytochrome switches back, and growth goes back to normal. And phytochrome is also involved with flowers. This is a greenhouse belonging to the French National Research Centre in Paris. In it, they've created an environment controlled for temperature, humidity, and light. Being able to turn day into night, botanists have found that plants can measure the length of day very accurately. And they use it to know when to flower. The African marigold flowers in the autumn. It can measure the shortening days. The summer-flowering pink dianthus won't appear until the days are more than 12 hours long. Some plants can work to accuracies of only 5 minutes. So how do they measure time? Plants, like most living organisms, have an internal clock which is set at roughly 24 hours. This bean plant has so-called sleep movements. Every 24 hours, it'll drop its leaves, even in constant daylight. We now know that in order to flower, plants measure not day length, but night length. Just a short flash of light at night may completely disrupt flowering. Experiments show the plant's light sensor is again phytochrome, the red sensitive pigment. So it and the plant's internal clock interact in the flowering process. But do we know how?
Non, on ne connaît pas bien et même presque pas du tout le mécanisme d'action de ce.
We've actually very little idea of how phytochrome works. We have no satisfactory explanation of what the plant's internal clock is, and that needs some working on.
Pas très bien quelle est l'horloge de la plante, et c'est ce qu'il faut rechercher actuellement.
Flowering isn't the only timing mechanism in plants. Another internal clock has been found in climbers. This is a bean shoot looking for a support. Each revolution takes about an hour and a half. Botanists in France have restricted the movement of 3 separate bean plants by taping the shoots at different heights. As the shorter shoots grow, they gradually revert to the same 90-minute cycle, and the 3 plants end up in perfect synchronization. Why and how this sense of timing occurs, no one knows. But we do know about the climbing mechanism itself.
The plant is sweeping through space blindly until it encounters a potential support. It bounces or slightly rubs against that support, and in fact, the cells on the surface of the tendril or the bean plant have little ridges that catch and rub against the support. And it's this rubbing that gives the sensory information to the plant and tells it to start coiling around the support.
In some plants, like the pea, this response is very rapid. As soon as its tendril has located a support, it starts to coil rapidly, sometimes within 20 seconds. Once attached, its original sweeping movements are ingeniously modified to pull it towards the support and provide natural independent suspension. How the pea tendril makes such rapid movements has been investigated by Professor Jaffe. He's removed tendrils and allowed them to take up a small dose of radioactive water. A couple of strokes and the coiling response is activated. Then comes the difficult bit. Cut the tendril right down the middle. How the coiling actually occurred will be shown by the levels of radioactivity on each side of the tendril. So, how do plants coil?
We've seen that water is flushed out of the cells on the underside of the tendril, and instead of going from these cells to these cells, moves out of the tendril and down into the plant. This loss of water in these cells makes them get smaller, much as a balloon would get smaller if it lost air. The pressure of the water in the cells on the upper part remains the same. This forces the tendril over. There has to be something going on within the cells that cause this. And interestingly enough, the kinds of mechanisms, the kinds of molecular mechanisms, may turn out to be very similar to those that are found in animal cells, specifically muscle cells. The kinds of molecules that are ultimately responsible for the contraction of a muscle and then for its relaxation are also found in these rapidly moving plant parts.
Perhaps the most famous plant muscle is in the Venus flytrap. It's certainly the most rapid, closing in a quarter of a second. Another rapid mover is the mimosa. Put a flame near one leaf and the whole plant will defend itself. the leaf drops by rapidly flushing water out of the cells at the base of the stalk. It takes less than 15 seconds to transmit the message around the plant. How does it work? Could it be due to a fast-acting hormone, or might it be something else? At Washington University in St. Louis, Dr. Barbara Pickard has been investigating. She's attached an array of electrodes to a tiny mimosa plant. She's hoping to pick up any electrical activity traveling through the plant. Each electrode is connected separately to a chart recorder. She pinches one leaf, and almost immediately the nearest electrodes respond. And then the other two. Electrical signals like these have also been found in the Venus flytrap. But how is the electricity generated? The answer is in the cell. It's a kind of battery. At Cambridge University, Dr. Enid Bagrobi.
Plant cells have different charged particles inside and out. Outside they have a low concentration of sodium chloride, and inside they have a much higher concentration of potassium chloride. And there is set up across the membrane, therefore, a difference in an imbalance of charged particles, which gives a membrane potential. And most plant cells would have a membrane potential of between 100 and 200 millivolts.
This is not the stem of a plant, it's one whole cell. It's the cell of a giant alga, and its size makes it ideal for research into plant electricity. At Cambridge, they've made a detailed study of the types of electrical output from the cell, and what it's produced brings plants and animals a little closer. A primitive nerve impulse.
The cell seems to be excitable in a way rather like a nerve action potential. If, for example, it's mechanically stimulated by pinching or a pressure change or a cold or a hot shock, then there is an action potential produced in which there's a spike and the potential falls over half a second perhaps and then recovers over a few seconds. And this change, which is very like a nerve action potential but about 1,000 times slower, can propagate along the cell and can also propagate from cell to cell.
This is the giant alga under the microscope, and this is what happens when it's stimulated. The moving contents of the cell stop, producing the nerve-like reaction.
In a primitive system like the giant algae, it's very difficult to see that this has any real function, but it suggests there is perhaps a basic means of communication in all cells, plant or animal, and that the system is the same, although the speed of transfer may be very different.
So do plants have a kind of primitive nervous system? In America, Barbara Pickard was curious to see if there was any natural electrical activity within quite ordinary plants, like this morning glory. The electrodes in position, the plant wasn't touched. It was left completely alone with only a chart recorder for company. At first, the results were disappointing. The plant seemed to be electrically dead. But then, something happened. Days of monitoring and yards of chart recordings later, Pickard had what she was looking for, proof of spontaneous electrical activity in the plant. Much of it seemed too random to be interpreted, but every now and again the plant produced bursts of nerve-like pulses. What did she think they were?
It's very difficult to assign significance to these regular trains of signals. We have no way of demonstrating now whether they are the result of some kind of biochemical action or indeed whether they are true signals. I find it very hard to believe that repetitive trains of signals which may last for several hours and which have such a distinctive neuroid waveform, such a rapid rise time, often as little as 1 millisecond, could be anything but signals.
Whether these traces are the evidence of a nervous system in plants isn't yet clear. The research is in its infancy, and some botanists are skeptical about its meaning. But there's another whole set of theories about plants that they really won't accept. In the last few years, a flurry of books have appeared claiming that plants have extraordinary powers of awareness. Plants, it is said, have extrasensory perception. A leading architect of the new theory is Cleve Baxter. He's not a botanist, but in fact is one of America's foremost experts in lie detection. His interest in plants began 10 years ago, when in an idle moment he decided to hook up his lie detection apparatus to a plant. He chose a philodendron, and to one leaf attached the lie detector electrodes. They work by passing a small current between them. On the skin of people, changes in the resistance between the electrodes are used to indicate emotional arousal. But what about plants? Baxter was surprised to find that the plant produced traces like this.
When I saw a contour of a reaction that looked very similar to a typical galvanic skin response in a human, I thought, my goodness, could this plant be showing me human-like reactions contour-wise? And I thought, well, with a human in a lie detection situation, you're actually measuring an emotional stimulation that ties into a threat to their well-being if they're lying to a very serious question. And so I thought, well, what do you do with a plant that's a threat to their well-being? And it suddenly occurred to me, almost out of the blue, I know what I'll do. I'll burn that leaf, that very leaf that I have attached. And the split-second timing of the time that the imagery of fire entered my mind and the reaction on the plant was very, very persuasive to me. I had no matches in the room or anything, and the thing just leaped up right off the top of the page and went into real wild agitation, and it had been very calm before that.
This is the record of that first experiment. He claimed the plant had picked up his threatening thoughts. Since then, he's taken recordings from a plant while threatening other life forms, ranging from an egg to his own cat. He concluded that plants could remotely detect injury to other biological organisms. In 1968, he did his first controlled experiment. He took tiny aquarium shrimps and dropped them at random intervals into boiling water. The recording he obtained from the plant convinced him that the plant could detect the death of the shrimps. It was a stunning claim. Other laboratories rushed to repeat the experiment, but to date, no one has succeeded in replicating it. But Baxter has not been daunted. His researches on plants have continued, and he offered Horizon a demonstration. This closed-circuit television system uses a split-screen device which shows the experimental area on the top and the simultaneous output from the plant on the bottom. The electronic link cannot be faked.
Now, first I'm going to try to find a spot where I can do a little cutting here.
He first tried the plant's reaction to the death of some of his own body cells.
See if I can get some
interaction here. I'm now starting to feel the cut as I'm getting under the surface a little bit. I want to get it opened enough to where I can get some iodine or iodide in the cut. Here we go. I think now that's getting some death of cells. And I will now do a little more cutting here. The skin is tough there. Okay, now I'm going to now try to work some iodide in there So I can start to feel the... Actually, this is iodine. I see it's nice and red. See if I can get this down into the puncture. There, we can start to feel it now. Here we go. There we go.
His conclusion?
The plant has rather consistently, at least observationally speaking, reacted to the death of human cells.
A second test. Injury to the cells of another plant. Radishes were to be ripped apart in a mixer.
Already? Oops, I forgot one little thing.
For Baxter, this produced a significant result.
Now, what I was doing then is trying to impulsively come over without any prior notification of the vegetable I was going to pick up. And I noticed even as I grabbed toward the radishes, it started to react already. Then I shoved them in there, and we got the action that you got here.
Here is an action replay of that event. The tracing from the plant, he said, had risen at the moment of his intention to injure the vegetable. We invited a botanist to come and investigate the experiments for himself. Professor David Rail of San Diego State University is a plant hormone specialist, and he gave the apparatus a thorough check.
Although these clamps are awfully tight, and I suspect that there is some cellular leakage which would change the resistance across these. You can see that rather. I think if you loosen and tighten these, you can see that there's a reasonably dramatic pen response. Have I got that too tight or too loose?
No, it's okay now. I have to rebalance it in, so that is it firmly.
All right, the point is whether we can reproducibly demonstrate some response from the plant as a result of some kind of threat. And the sort of threat that we've chosen is to anticipate plunging this particular plant into very hot water. And I will anticipate plunging it in, and then I will plunge it in. And I'm thinking about it now, and in it goes. The hot water should kill a number of cells, although probably not all of them. And I will now take it out, and you can see that cells are very limp. Obviously, we've injured some cells and lost considerable turgor from these cells. Now, I don't know whether that initiated a response or not.
No response. The response comes when you saw that it didn't initiate a response. Which was
as a control, in anticipation of some kind of response, I anticipate plunging dry leaves into the same beaker, so I'll do that and see if perhaps this initiates a response while the live plant didn't. Well, let's let those soak there for a moment. Now, did that initiate a response?
No, unless there were bacteria or something. I wouldn't expect.
So Professor Rail tried some bacteria. Yogurt.
I'm going to take a spoonful of yogurt and drop it into the boiling water.
Here's the action replay.
I'm going to take a spoonful of yogurt and drop it into the boiling water.
For Cleve Baxter, the rise in the trace showed the plant had picked up Dr. Rail's intention of harming the yogurt. But Rail himself wasn't convinced.
You see some deflection of the pen, but it's not apparent that it's actually due to any event that I initiated.
But he does think that Baxter ought to pursue the work, though in a much more scientific way.
The problem, of course, is that in any situation like this, what you're observing is an event. It's not an experiment. It's an observation. In all of these experiments, there is no control. That is, there is no inanimate object attached, a dead leaf, a piece of cellulose, a piece of filter paper attached to a similar polygraph. So we have no way of knowing whether the events that we see are due to environmental fluctuations, fine voltage fluctuations, or simply random discharge from the cells, if really that's what we're measuring.
To date, Baxter's work has been rejected by botanists the world over.
I think skepticism in the scientific community is extremely healthy. I've been very low-key in stating that I don't feel that I've proven anything, but as a person that's had 28 years, in excess of 28 years experience in reading psychophysiological tracings from people, that when I'm hooking up plants and some of the other life forms that I am testing, I'm seeing some very interesting tracings that seem meaningful to me. And I'm only suggesting that they look. I'm not suggesting that they believe.
Whether plants have extrasensory perception remains unproven, but it's not surprising the idea has caught on. After all, the known powers of plants are pretty extraordinary, and many mysteries still remain. Plants haven't always attracted the greatest scientific interest. Perhaps they should, for without them, we wouldn't be here.